Hello there, and welcome to the Sleepy Science Society. Tonight, we're drifting into a world that exists all around us, yet remains mostly invisible to our hurried eyes. A world of silent battles, chemical weapons, and strategic alliances that would make any military general envious. We're exploring the extraordinary defense mechanisms of plants, and I promise you, by the end of this journey, you'll never look at a simple garden the same way again. Before we begin this peaceful exploration, if you enjoy these quiet journeys through science, I invite you to like this video and subscribe to the channel. It helps others find their way here too, one sleepy soul at a time. But for now, all you need to do is settle in, let your breathing slow, and prepare to see the botanical world through entirely new eyes. Imagine for a moment that you are rooted to one spot. You cannot run from danger. You cannot hide when threatened. You must face every predator, every disease, every challenge exactly where you stand. This is the reality for every plant on Earth. And yet, they thrive. They've colonized nearly every habitat on the planet. They've survived for hundreds of millions of years. How? Through some of the most sophisticated, elegant, and sometimes ruthless defense systems ever evolved by living organisms. Plants are not passive victims. They are active defenders, equipped with an arsenal that would astound any weapons designer. They produce thousands of different chemical compounds, some so toxic they can kill a large animal within hours. They've developed armor plating in the form of spines and thorns. They've created communication systems that warn their neighbors of approaching danger. Some have even formed military alliances with insects, trading food for protection. And perhaps most remarkably, they can sense when they're being attacked and mount coordinated defense responses within seconds. Tonight, we'll journey through this hidden world of botanical warfare. We'll discover how a simple blade of grass can be as well defended as a fortress. We'll learn about plants that can hear caterpillars chewing and respond by calling in aerial support. We'll explore the underground networks that allow trees to share information about threats sweeping through the forest. And we'll witness some of the most bizarre adaptations in nature, where plants have turned the tables completely and become the hunters themselves. So let your mind drift gently into this verdant battlefield. Allow yourself to shrink down to the size of an insect, where a single leaf becomes a landscape, and a flower becomes a complex structure of chemistry and architecture. In this miniature world, every surface tells a story of evolution, adaptation, and survival. The gentle swaying of branches in the breeze takes on new meaning when you understand the chemical signals floating through that air. The simple act of a plant growing becomes an act of strategic resource allocation, deciding how much energy to invest in growth versus defense. As we explore together, you might find your eyelids growing heavy. That's perfectly fine. Let the information wash over you like a gentle stream. Some details will catch and settle into your memory. Others will simply flow past, creating a soothing rhythm of fascinating facts. There's no test at the end. No need to remember everything. Just allow yourself to experience the wonder of it all, to feel a growing appreciation for the complexity and beauty of the plant kingdom that surrounds us every day. The ancient arms race. The story of plant defenses begins hundreds of millions of years ago in a world vastly different from our own. When plants first emerged from the water to colonize the land, roughly 450 million years ago, they entered a realm of new possibilities and new dangers. The land offered abundant sunlight and carbon dioxide, resources that could fuel unprecedented growth. But it also exposed these pioneering plants to new threats: drying winds, intense ultraviolet radiation, and eventually the animals that would follow them onto land and begin eating them. The first land plants were simple organisms, little more than mats of green tissue hugging moist surfaces. They had no roots, no leaves in the modern sense, no flowers or seeds. But even these primitive plants faced herbivory. Tiny arthropods, the ancestors of modern insects and millipedes, began feeding on these early terrestrial plants. And so began one of the longest and most consequential arms races in evolutionary history. An arms race in evolution occurs when two groups of organisms exert selective pressure on each other, driving continuous adaptation and counter-adaptation. When herbivores evolved better ways to eat plants, natural selection favored plants that developed better defenses. When plants developed those defenses, natural selection favored herbivores that could overcome them. Back and forth, generation after generation, each side pushing the other to evolve new strategies, new weapons, new tactics. Over the vast stretches of geological time, this evolutionary arms race has produced diversity beyond imagination. Scientists estimate there are approximately 300,000 plant species on Earth today, and each one represents a unique collection of defense strategies. Some plants have invested heavily in physical defenses, growing spines so sharp and dense they're nearly impossible to eat. Others have pursued the path of chemical warfare, producing the compounds so bitter, so toxic, or so confusing to herbivore nervous systems that animals quickly learn to avoid them. Consider the sheer creativity of these adaptations. Some plants produce chemicals that mimic insect hormones, disrupting the development of any caterpillar that tries to eat them. The unfortunate larva might pupate too early or fail to pupate at all, its body confused by signals that shouldn't be there. Other plants produce compounds that make them nutritionally worthless, filling an herbivore's stomach without providing any usable nutrients, essentially starving the animal even as it eats. The pace of this evolutionary race has varied over time. During the Cretaceous period, roughly 140 to 65 million years ago, something remarkable happened. Flowering plants, the angiosperms, underwent an explosive diversification. In geological terms, they appeared suddenly and spread rapidly, prompting Charles Darwin to call their origin an abominable mystery. While many factors contributed to this success, their sophisticated defense systems played a crucial role. Flowering plants developed new types of chemical defenses that were particularly effective against the insects that dominated terrestrial herbivory. They created complex molecules that required specialized metabolic pathways, molecules that weren't found in their ancestors. These secondary metabolites, as scientists call them, served no direct role in basic plant functions like photosynthesis or reproduction. Instead, they existed purely for defense, for warfare, for survival. But here's where the story becomes even more fascinating. As plants diversified their defenses, insects evolved countermeasures. Some herbivorous insects developed the ability to detoxify plant poisons, turning the very toxins meant to kill them into harmless waste products. Others went further, sequestering plant toxins in their own bodies, storing these poisons to make themselves toxic to their own predators. The monarch butterfly, feeding on toxic milkweed plants and storing those toxins to make itself poisonous to birds, represents one of the most elegant examples of this strategy. This created a pattern ecologists call escape and radiation. A plant lineage would evolve a novel defense, perhaps a new type of toxic compound. This innovation would provide temporary escape from herbivores, allowing that plant lineage to diversify rapidly into new species. Eventually, some insects would evolve the ability to overcome this defense, and they too would diversify into new species specialized on these previously protected plants. The pattern would then repeat, driving ever greater diversity in both plants and insects. The fossil record provides glimpses of this ancient arms race. Paleobotanists have discovered leaves from ancient plants showing evidence of insect-feeding damage, the same types of holes and nibbled edges we see today. They've found fossilized insect mouthparts adapted for piercing plant tissues. They've even found plant fossils that show structural defenses like spines and tough tissues. The war between plants and herbivores isn't just happening now, it's been raging for hundreds of millions of years, and the modern forest or meadow represents simply the latest chapter in this epic struggle. In this ongoing conflict, neither side has achieved complete victory. Plants haven't evolved defenses so perfect that no animal can eat them. If they had, herbivores would have gone extinct long ago. Conversely, herbivores haven't overcome all plant defenses. If they had, plants would have been eaten to extinction. Instead, we see a dynamic equilibrium, a constant back and forth, where both sides remain in the game, both continuing to evolve, both locked in an embrace of mutual influence that shapes the very structure of ecosystems. Understanding this historical context helps us appreciate what we're about to explore. Every defense mechanism we'll discuss tonight represents a solution to a problem millions of years old. Every chemical weapon, every physical barrier, every clever strategy exists because somewhere in the deep past, a plant lineage that possessed it survived better than its neighbors. These aren't random features. They're the accumulated wisdom of eons, written in the language of DNA, expressed in the structures and substances we can observe today. Physical barriers, the first line of defense. When you reach out to touch a rose, the thorns on its stem serve as an immediate reminder that plants are not defenseless. These sharp protrusions represent one of the most ancient and straightforward defense strategies in the plant kingdom. Make yourself physically difficult or painful to eat. Physical defenses, sometimes called mechanical defenses, form the first line of protection for countless plant species. They're present all the time, ready to work whether an herbivore approaches or not. And they come in a remarkable variety of forms, each shaped by the particular challenges faced by different plants. Let's start with the most obvious and familiar: thorns, spines, and prickles. While we often use these terms interchangeably in everyday language, botanists distinguish between them based on what part of the plant they're modified from. True thorns are modified branches. They develop from the plant's vascular tissue and contain all the structural elements of a stem, just miniaturized and sharpened to a point. The thorns on hawthorn trees or citrus plants fall into this category. When you see a thorn, you're looking at what is essentially a weaponized branch, a structure that could have grown into a leaf-bearing stem, but instead became a defensive spike. Spines, on the other hand, are modified leaves or parts of leaves. Cacti provide the most familiar examples. Those sharp points covering a cactus aren't just decorative. They're leaves that have been completely transformed for defense and water conservation. In the harsh desert environments where cacti evolved, broad leaves would lose precious water through transpiration. By converting leaves into spines, cacti solved two problems at once. They reduced water loss and protected themselves from herbivores desperate for moisture in arid landscapes. Then we have prickles, which are often confused with thorns but have a different origin entirely. Prickles are outgrowths of the plant's epidermis, the outer cell layer. They're not connected to the vascular system and can be removed relatively easily, taking only surface tissue with them. Roses actually have prickles, not thorns, though we commonly call them thorns. Blackberries and raspberries also sport prickles along their stems, making them unpleasant to brush against and tedious to harvest, as anyone who's picked wild berries can attest. The effectiveness of these sharp structures lies not just in their ability to cause pain, but in their ability to make feeding more difficult and time-consuming. An herbivore that must carefully navigate around thorns or spines spends more time exposed to predators. It expends more energy for the same amount of food. Over time, many herbivores learn to avoid heavily defended plants entirely, seeking out less troublesome food sources. Natural selection thus favors individuals with effective physical defenses. But physical defenses extend far beyond obvious spikes and points. Many plants are covered in trichomes, tiny, hair-like structures that protrude from their surface. At first glance, these might seem insignificant, perhaps serving only to give a plant a fuzzy appearance. But trichomes represent a sophisticated defense system with multiple functions. Some trichomes are simple and stiff, creating a physical barrier that makes it difficult for small insects to reach the plant's surface. Imagine trying to walk through a dense forest of rigid spikes, each one proportionally as tall as you are. That's what a caterpillar faces when trying to crawl across a heavily trichomed leaf. Other trichomes have evolved even more elaborate defenses. Glandular trichomes contain specialized cells that produce and secrete defensive chemicals. In plants like tomatoes and certain mints, these glandular hairs manufacture sticky resins or toxic compounds. When an insect brushes against these trichomes, the secretions coat its body, gumming up its legs and mouth parts. Small insects can become completely immobilized, effectively glued to the plant surface. The trichomes of stinging nettles take this even further, acting like hypodermic needles that inject irritating chemicals when touched, causing the familiar burning sensation that can last for hours. The diversity of trichome forms and functions across the plant kingdom is astonishing. Some are hooked, catching on the legs and mouth parts of insects. Others are branched, creating an even more impenetrable barrier. Scientists have discovered that the density and type of trichomes on a plant's surface can vary depending on environmental conditions and the presence of herbivores. Some plants actually increase their trichome production in response to attack, adding another layer of defense after initial damage occurs. Moving beneath the surface, we encounter another crucial physical barrier: the cuticle. This waxy layer coating the epidermis of leaves and young stems serves many functions, including reducing water loss. But it also provides defense. The cuticle creates a tough, slippery surface that is difficult for fungi to penetrate and challenging for some insects to grip. Its waxy composition also makes it resistant to certain digestive enzymes that herbivores might secrete. Plants in harsh environments often have particularly thick cuticles, reflecting the dual challenges of water stress and herbivore pressure. Beneath the cuticle, the cell wall itself represents another physical barrier. Plant cell walls are made primarily of cellulose, a complex carbohydrate that's difficult to digest. Most animals lack the enzymes necessary to break down cellulose efficiently. Those that do eat plants extensively, like cows and termites, rely on symbiotic microorganisms in their digestive systems to break down cellulose for them. But some plants go further, reinforcing their cell walls with lignin, the compound that makes wood hard. Lignification, as this process is called, turns soft tissue into something much tougher and less palatable. We see this clearly in the progression of a plant through its growing season. Young leaves are often tender and easily eaten, their cell walls thin and unlignified. As leaves mature, many plant species increase lignin deposition, making the leaves progressively tougher. Herbivores, given a choice, consistently prefer younger leaves. The toughness of mature leaves isn't accidental. It's a defense strategy, a way of reducing losses once leaves have completed their most active growth phase. Trees take lignification to an extreme with bark. The outer bark of a tree represents one of the most effective physical barriers in nature. Thick, tough, sometimes nearly impenetrable, bark protects the vital living tissues underneath from a wide array of threats: insects, fungi, fire, physical damage, and temperature extremes. Different tree species have developed remarkably different bark characteristics, reflecting their particular evolutionary pressures. Some have smooth, thin bark that's continually shed, taking any colonizing organisms with it. Others develop thick, furrowed bark that creates a stable surface, but with such tough tissue that penetrating it requires specialized adaptations. Consider the cork oak, whose bark can be harvested repeatedly without killing the tree because it grows such an extraordinary thickness of protective tissue. Or think of the giant sequoias, whose bark can exceed a foot in thickness, providing protection from the fires that regularly sweep through their native habitats. The bark of these ancient trees contains so much insulating material that moderate fires pass by without damaging the living cambium beneath. Some plants deploy physical defenses that are less obvious but equally effective. Grasses, for instance, incorporate silica into their tissues, creating microscopic, glass-like structures called phytoliths. These make the leaves abrasive and difficult to chew, wearing down the teeth of herbivorous mammals. Over evolutionary time, grazing, mammals responded by developing ever-growing teeth that continuously replace tissue worn away by silica-rich grass. This represents another example of the evolutionary arms race we discussed earlier, played out in dental anatomy. Certain plants produce specialized structures that break off inside wounds, making injuries more severe and potentially leading to infections. The spines of some cacti, for instance, have microscopic barbs that cause them to penetrate deeper when an animal tries to remove them. Other plants have brittle hairs that break off and embed in the mouth or digestive tract of herbivores, causing irritation and discomfort. We should note that maintaining all these physical defenses comes at a cost. Producing thorns, spines, thick cuticles, and tough cell walls requires resources, energy, nutrients, and time. A plant that invests heavily in physical defense is a plant that has fewer resources available for growth or reproduction. This leads to interesting patterns in nature. Plants in resource-rich environments, where herbivore pressure is high but resources for regrowth are abundant, often invest less in permanent physical defenses. Plants in resource-poor environments, where replacing lost tissue is expensive, typically invest more heavily in defensive structures. As we consider these physical barriers, from the obvious thorns on a rose to the microscopic silica in grass, a picture emerges of plants as active architects of their own protection. They're not passive victims waiting to be eaten. They are sophisticated organisms that have invested millions of years and countless generations into developing structures that keep them alive in a world full of creatures trying to consume them. And, as effective as these physical defenses are, they represent only the first layer of protection. Behind this physical fortress lies an even more sophisticated system of chemical defenses that we'll explore next. Chemical warfare, secondary metabolites. If physical defenses represent the armor of plants, chemical defenses represent their weapons. And what weapons they are. Plants produce tens of thousands of different chemical compounds, many of which have profound effects on the animals that encounter them. Some of these chemicals are mildly unpleasant, creating bitter tastes or bad odors that discourage feeding. Others are potently toxic, capable of killing herbivores quickly if consumed in sufficient quantities. Still, others are more subtle, disrupting the physiology or behavior of herbivores in ways that reduce their fitness without necessarily killing them outright. These defensive chemicals are called secondary metabolites, distinguishing them from primary metabolites like sugars, amino acids, and nucleic acids that are essential for basic life processes in all organisms. Secondary metabolites serve specialized functions, and in many cases, that function is defense. The diversity of secondary metabolites produced by plants is staggering. Scientists have identified more than 50,000 different compounds, and this number grows each year as we discover new molecules in previously unstudied plants. Let's explore the major categories of these chemical weapons, understanding how they work and appreciating the elegant solutions to survival challenges that they represent. We'll begin with perhaps the most well-known class. Alkaloids. The name comes from their chemistry. They're basic compounds with properties opposite to acids, but their significance comes from their effects on animal nervous systems. Alkaloids are nitrogen-containing compounds that interact with nervous system function in various ways. Caffeine, the compound that makes your morning coffee stimulating, is an alkaloid. So is nicotine, the addictive compound in tobacco. Morphine, the powerful painkiller, is an alkaloid derived from poppies. These examples immediately reveal something important. Alkaloids can be potent. They cross into the brain and affect neural function, sometimes profoundly. From the plant's perspective, this makes alkaloids excellent defenses. Herbivores that consume alkaloid-rich plants often experience unpleasant effects: nausea, confusion, increased heart rate, tremors, or in extreme cases, paralysis and death. The bitter taste of many alkaloids provides an initial warning, causing many animals to reject alkaloid-containing plants after a single nibble. Those that persist in eating such plants may suffer consequences that teach them, through memorable discomfort, to avoid those plants in the future. Consider strychnine, one of the most toxic alkaloids known. Produced by the seeds of the Strychnos nux vomica tree, strychnine interferes with the nervous system's inhibitory signals, causing violent muscular contractions and convulsions. A tiny amount can be lethal to humans. The tree doesn't produce strychnine to harm humans specifically, of course, but any animal that tries to eat those seeds faces the same deadly response. Over generations, animals learn, either individually or through instinctive aversions evolved over time, to leave those seeds alone. Solanine, found in potato plants, particularly in green potatoes and potato sprouts, provides another example. This alkaloid serves as a defense against fungi and insects. In humans, consuming significant amounts causes gastrointestinal distress, headaches, and neurological effects. The distinctive, bitter taste of solanine serves as a warning, prompting most people to avoid green potatoes instinctively. Moving to another major class of defensive chemicals, we encounter terpenes. These compounds are built from basic 5-carbon units that can be assembled into an enormous variety of structures. Some terpenes are small and volatile, evaporating easily into the air. Others are large and complex, remaining within plant tissues. Many terpenes have strong odors, and indeed, terpenes are responsible for the characteristic scents of many plants. The fragrance of pine trees comes from terpenes called pinene. The smell of lavender, rosemary, and mint. Terpenes, the distinctive aroma of cannabis, a complex blend of terpenes. These scents aren't just pleasant to humans. They often serve defensive functions, either repelling herbivores directly or serving as signals in more complex defense strategies. Some terpenes are directly toxic to insects. Pyrethrin, derived from chrysanthemum flowers, is a terpene that attacks the nervous systems of insects, making it an effective natural insecticide. In fact, pyrethrins were some of the first plant-derived compounds used commercially for pest control. Other terpenes interfere with insect hormone systems, disrupting development and reproduction. Plants in the mint family produce particularly diverse and potent terpene mixtures. If you crush a mint leaf between your fingers, The strong smell that's released comes from specialized structures called oil glands rupturing and releasing their volatile terpene contents. This same mixture that smells refreshing to us can be irritating or even toxic to many insects. The volatile terpenes can also act as airborne signals, a topic we'll explore more deeply when we discuss plant communication. Phenolic compounds represent another major category of chemical defenses. These molecules contain one or more hydroxyl groups attached to an aromatic ring, a structure that gives them distinctive properties. The simplest phenolic compounds serve as building blocks for more complex molecules, including lignin, which we mentioned earlier in the context of physical defenses. But phenolic compounds also function as chemical weapons in their own right. Tannins, for example, are complex phenolic compounds found in high concentrations in oak leaves, tea leaves, and many other plants. If you've ever tasted very strong tea that was steeped too long, you've experienced the astringent, mouth-puckering effect of tannins. This astringency comes from tannins binding to proteins in your saliva, causing them to precipitate. This same protein-binding property makes tannins effective defenses. When an herbivore eats tannin-rich leaves, the tannins bind to proteins in the animal's digestive system, reducing the nutritional value of the meal. In high concentrations, tannins can cause digestive problems, essentially making the plant very difficult to digest and derive nutrition from. Some herbivores have evolved tannin-tolerant digestive systems, with special proteins that bind tannins before they can interfere with digestion. But for many potential herbivores, tannin-rich plants are simply not worth eating. Some phenolic compounds go beyond interference with digestion to become directly toxic. Gossypol, found in cotton plants, is a phenolic compound that can damage liver and heart function in many animals. Juglone, produced by walnut trees, is toxic to many plant species, serving as a form of chemical warfare against competing plants that might grow too close. This compound also affects animals, particularly horses, which can develop severe conditions if exposed to black walnut wood. Now we come to one of the most dramatic categories of plant defenses: cyanogenic glycosides. These compounds, when damaged or chewed, release hydrogen cyanide, one of the most rapidly acting poisons known. Cyanide blocks cellular respiration at a fundamental level, preventing cells from using oxygen. In high enough doses, this causes rapid death. Plants that use cyanogenic glycosides store them safely within their tissues using a clever two-part system. The cyanogenic glycoside itself is stored in one cellular compartment, while the enzyme that releases cyanide from it is stored separately. When an herbivore chews the plant tissue, these compartments break open, the compounds mix, and cyanide is released. It's like a biochemical landmine, harmless until triggered by damage. Cassava, a staple food crop in many tropical regions, contains high levels of cyanogenic glycosides. Traditional food preparation methods, including soaking and fermenting the roots, are specifically designed to break down these compounds and allow the cyanide to evaporate before consumption. Improper preparation can lead to cyanide poisoning. The wild cherries that dot many North American forests also produce cyanogenic glycosides, particularly in their wilted leaves. There are documented cases of livestock dying after eating fallen cherry leaves. Beyond these major categories, plants produce countless other specialized defensive compounds. Glucosinolates in the mustard family give vegetables like broccoli and cabbage their distinctive flavors, and also serve as defenses against many insects. When plant tissue is damaged, glucosinolates break down to form compounds that can be toxic or repellent to herbivores. Latex, the milky fluid that oozes from dandelions, milkweeds, and rubber trees when damaged, serves both as a physical and chemical defense. It's sticky, potentially gumming up the mouthparts of insects. But it also contains various toxic or deterrent compounds. The white sap from milkweeds contains cardiac glycosides, compounds that affect heart function and can be poisonous to vertebrates. Some plants produce compounds that are primarily deterrent rather than toxic. They taste terrible, smell bad, or create unpleasant sensations without necessarily causing serious harm. Capsaicin, the compound that makes chili peppers hot, falls into this category for mammals. Interestingly, birds are insensitive to capsaicin, allowing them to eat chili peppers and disperse the seeds while mammals are deterred from eating them. This represents a sophisticated evolutionary strategy. Deter seed predators. Mammals that would destroy the seeds with their powerful jaws and complex digestive systems, while allowing seed dispersers, birds, free access. The production of all these chemical defenses requires significant resources. Plants must synthesize complex molecules using energy from photosynthesis and nutrients from the soil. They must store these compounds safely, often in specialized structures, to avoid poisoning themselves. And they must maintain these chemical arsenals even when herbivores aren't present, because attacks can come at any time. This cost leads to interesting patterns in plant chemistry. Plants growing in nutrient-rich environments, where replacing lost tissue is relatively easy, often invest less in chemical defenses than plants in nutrient-poor environments. Young, rapidly growing tissues are often less well-defended chemically than mature tissues, because the cost of defense competes with the cost of growth, and young tissues can be replaced more easily. Seasonal patterns in chemical defense are also common. Many plants produce higher concentrations of defensive chemicals during times when herbivore pressure is highest, such as spring, when insects are abundant and active. Some plants even show circadian rhythms in their defensive chemistry, with concentrations of certain compounds varying throughout the day and night. As we contemplate this vast chemical arsenal, spanning thousands of different compounds with diverse modes of action, we begin to appreciate plants not as passive fixtures of the landscape, but as sophisticated chemical laboratories. Every leaf, every stem contains a complex mixture of molecules, fine-tuned by millions of years of evolution to deter, poison, or confuse the animals that would eat them. And, remarkably, this chemical warfare system isn't static. Plants can activate and enhance their chemical defenses dynamically in response to attack, a phenomenon we'll explore next. Induced defenses. The awakening response. Imagine having a home security system that only turns on when you detect an intruder, saving energy and resources the rest of the time. This is essentially what many plants do with their induced defense systems. Rather than maintaining maximum defensive capabilities at all times, which would be energetically expensive, these plants can detect when they're being attacked and rapidly mobilize additional defenses specifically in response to that threat. It's an elegant solution to a fundamental problem: how to be well defended without paying the constant cost of maintaining those defenses. The story of induced defenses begins with perception. A plant must somehow detect that it's being attacked before it can respond. You might wonder how a plant, lacking eyes, ears, or a nervous system, could possibly detect an herbivore. The answer lies in the damage itself and the chemicals that herbivores introduce while feeding. When an insect chews on a leaf or a pathogen invades tissue, it causes physical damage that plant cells can detect. Moreover, the saliva of many herbivorous insects contains specific chemicals that plants have evolved to recognize as danger signals. The moment that damage occurs, a cascade of events begins inside the plant. Damaged cells release molecules that act as alarm signals. Calcium ions begin flooding into cells, creating electrical signals that propagate through the plant tissue. Within seconds, plant cells dozens of centimeters away from the damage site can begin responding, even though they haven't been touched by the herbivore yet. The speed of these initial responses approaches that of neural transmission in animals, though the mechanisms are entirely different. At the heart of the induced defense system is a hormone called jasmonic acid, or JA for short. When a plant detects herbivore damage, one of the first responses is a rapid increase in J levels. This hormone acts as a master regulator, triggering changes in gene expression throughout the plant. Within hours of an attack, thousands of genes may be activated or suppressed in response to JA signaling. These genes encode a diverse array of defensive proteins and enzymes. Some of the proteins produced in response to JA are enzymes that synthesize defensive chemicals. Remember those secondary metabolites we discussed earlier: alkaloids, terpenes, phenolics. Many of these are not present at high levels in undamaged plants. Instead, the enzymes that make them are ramped up after attack begins. The plant essentially shifts its metabolism into war mode, producing defensive compounds at accelerated rates. Other proteins induced by JA directly interfere with herbivore feeding. Protease inhibitors, for example, are proteins that block the action of digestive enzymes in an herbivore's gut. When an insect consumes leaves containing high levels of protease inhibitors, it cannot properly digest the proteins it's eating, essentially starving even as it continues to feed. The insect's growth slows, it takes longer to develop, and it becomes more vulnerable to predators and diseases. From the plant's perspective, producing protease inhibitors is cheaper than producing highly toxic compounds, yet the effect can be nearly as protective. Polyphenol oxidases are another class of defensive proteins induced by herbivore attack. These enzymes catalyze reactions that create dark indigestible compounds when plant tissue is damaged and exposed to air. If you've ever noticed an apple turn brown after you cut it or bite into it, you've seen polyphenol oxidases in action. While this browning might seem like mere oxidation, it actually makes the tissue less nutritious and less palatable to herbivores. Some plant species dramatically increase polyphenol oxidase production after being damaged, making subsequent bites even less appetizing. The induced defense response isn't limited to the site of damage. Through a process called systemic induction, the entire plant can become more resistant to herbivores after localized damage. The alarm signal from the damaged tissue spreads through the plant's vascular system, carried by the same channels that transport water and nutrients. As this signal moves through the plant, it activates defensive genes in tissues that haven't been touched. This means that if an insect starts eating one leaf, all the other leaves on the plant can become more defensive within hours or days. The speed and extent of this systemic response varies among plant species. Some plants show strong systemic induction, with defense-related compounds increasing throughout the entire plant within a day of initial damage. Others show more localized responses, with defenses increasing primarily in the damaged tissue and immediately adjacent areas. The pattern seems to relate to how mobile herbivores are and how likely they are to move from one part of the plant to another. One of the most fascinating aspects of induced defenses is that plants can sometimes distinguish between different types of attackers and tailor their responses accordingly. The saliva of caterpillars contains different chemicals than the saliva of, say, spider mites. Plants can detect these differences and activate different defensive strategies. Caterpillar damage might trigger the production of protease inhibitors and certain volatile compounds, while mite damage might trigger a different suite of defensive responses more effective against those particular herbivores. Even more remarkably, some plants can distinguish between mechanical damage, like a leaf being cut by wind-blown debris, and herbivore damage. A simple cut doesn't always trigger the full induced defense response, but damage combined with herbivore saliva does. This specificity prevents the plant from wasting resources responding to every minor injury, reserving the costly defense response for genuine threats. The phenomenon of systemic acquired resistance, or SAR, represents an even more sophisticated form of induced defense, primarily functioning against pathogens rather than herbivores. When a plant is infected by a pathogen in one location, it can develop enhanced resistance to subsequent infections throughout the entire plant and even to different types of pathogens. This is sometimes called immune memory in plants, though it operates through molecular mechanisms entirely different from animal immunity. SAR involves a different signaling molecule, salicylic acid, which is closely related to the active ingredient in aspirin. When pathogen infection occurs, salicylic acid levels rise throughout the plant, triggering the production of pathogenesis-related proteins. These proteins have various defensive functions, including directly attacking pathogen cell walls, strengthening plant cell walls, and producing antimicrobial compounds called phytoalexins. Phytoalexins are low molecular weight antimicrobial compounds that plants synthesize rapidly in response to infection or stress. Different plant families produce different phytoalexins, reflecting their diverse evolutionary histories. In the legume family, compounds like phaseolin and glyceolin serve as phytoalexins. In the potato family, it might be rishetin or phytuberin. These compounds accumulate to high concentrations at infection sites, creating a chemically hostile environment that can halt or slow pathogen growth. The induction of defenses allows plants to be strategic in their resource allocation. Maintaining maximum defensive capabilities at all times would require constant energy investment. By keeping defenses at a baseline level and ramping them up only when needed, plants can allocate more resources to growth and reproduction under peaceful conditions. When attack comes, they can temporarily reduce growth and invest heavily in defense, then ramp growth back up once the threat passes. However, this strategy isn't without costs and risks. There's a lag time between when attack begins and when induced defenses reach full effectiveness. An herbivore feeding during this lag period can cause significant damage. Additionally, repeatedly inducing defenses is still costly. Studies have shown that plants experiencing frequent herbivore attacks grow more slowly and produce fewer offspring than undamaged plants. Even when those attacks don't remove much tissue, simply because of the energetic cost of maintaining elevated defenses. The balance between constitutive defenses, always present, and induced defenses, activated only when needed, varies across the plant kingdom, reflecting different evolutionary solutions to the defense problem. Some plants rely heavily on constitutive defenses, maintaining high levels of toxins or tough physical barriers at all times. These tend to be long-lived plants in environments where herbivore pressure is consistently high. Other plants invest more in induced defenses, perhaps because they grow in variable environments where herbivore pressure is unpredictable, or because they're short-lived and need to minimize baseline costs. Climate and environmental factors also influence defense induction. Temperature, water availability, and nutrient availability all affect how strongly a plant can respond to attack. Drought-stressed plants, for instance, often show weaker induced responses because they lack the resources to mount strong defenses while also managing water stress. This can make stressed plants particularly vulnerable to herbivores, creating feedback loops where environmental stress leads to increased herbivore damage, which further stresses the plant. As we consider induced defenses, we're witnessing plants actively responding to their environment, making decisions about resource allocation, deploying specific counter-strategies against specific threats. These aren't conscious decisions, of course, but the result of millions of years of natural selection producing sophisticated regulatory systems. The plant that responds most effectively to herbivore damage, that mounts the right defense at the right time, is the plant that survives to reproduce. Over countless generations, this process has generated defense systems of remarkable complexity and effectiveness. Communication, networks, warning the neighbors. One of the most surprising discoveries in plant biology over the past few decades has been that plants don't defend in isolation. They communicate with each other, sharing information about threats, and this communication can help nearby plants prepare their own defenses before they're attacked. It's a finding that has fundamentally changed how we think about plants and their place in ecological communities. Rather than viewing forests and fields as collections of individual, isolated organisms, we now recognize them as interconnected networks where information flows between individuals in ways that benefit the community. The story of plant communication begins with volatile organic compounds, or VOCs. These are small molecules that evaporate easily into the air at normal temperatures. When a plant is attacked by herbivores, it releases specific blends of VOCs into the surrounding air. Initially, scientists thought these volatile releases were simply byproducts of plant damage or waste products from defensive metabolism. But careful experiments revealed something remarkable. These airborne compounds serve as signals that neighboring plants can detect and respond to. The first clear demonstration of this phenomenon came from studies on willow trees and wild tobacco plants. When researchers allowed herbivorous insects to feed on some plants while leaving nearby plants undamaged, they found that the neighboring plants, despite not being touched by herbivores themselves, began ramping up their defenses. Their leaves showed increased levels of defensive chemicals and enhanced resistance to herbivores. The plants were responding to something in the air, preparing for an attack that hadn't yet come to them but had befallen their neighbors. Further research identified the specific compounds involved. When a plant is damaged by herbivores, it releases a complex bouquet of VOCs, including compounds like greenleaf volatiles, aldehydes and alcohols that give cut grass its characteristic smell, terpenoids, and methyl jasmonate, a volatile form of the jasmonic acid hormone we discussed earlier. Different plants release somewhat different blends, but there's enough similarity that plants can recognize and respond to VOCs from both their own species and from other species sharing their environment. The responses triggered by these airborne signals mirror those triggered by direct damage. Neighboring plants that detect VOCs from damaged plants will increase their production of defensive enzymes, accumulate higher levels of secondary metabolites, and prepare to defend themselves more vigorously. In essence, they are priming their defenses, getting ready for trouble. When herbivores do finally reach these forewarned plants, they encounter much stiffer resistance than they would have otherwise. This raises fascinating evolutionary questions. Why would a plant warn its neighbors? If you're being attacked by caterpillars, doesn't it benefit you if those caterpillars move to neighboring plants instead? From a purely individualistic perspective, attracting herbivores to your neighbors seems like it would be advantageous. Yet plants do signal each other, and neighboring plants do benefit from these signals. Several hypotheses attempt to explain this apparent altruism. One possibility is that the VOCs aren't primarily signals to other plants, but rather serve other defensive functions, with the communication being a side effect. For instance, some VOCs directly repel herbivores, or attract the predators and parasites of herbivores. The fact that neighboring plants can eavesdrop on these signals might be incidental to their primary function. Another hypothesis suggests that kin selection plays a role. In many natural settings, neighboring plants are genetically related, having grown from seeds produced by the same parent plant, or even being part of the same clonal individual connected by underground stems. Warning your genetic relatives helps propagate your genes, even if it provides no direct benefit to the signaling individual. In such cases, communication benefits the signaler indirectly through effects on kin. A third possibility is that plants benefit from living among well-defended neighbors. If all the plants in an area are difficult to eat, herbivores might move away from that area entirely, reducing overall herbivore pressure on all individuals. It's a community-level defense strategy, where cooperation produces collective benefits that individual defense alone couldn't achieve. Whatever the ultimate evolutionary explanation, the phenomenon itself is well established and ecologically important. Farmers and gardeners can even exploit this communication. Interplanting susceptible crops with plants that naturally produce strong VOC signals can help protect the crops through this neighbor warning effect. But airborne signaling is just one avenue of plant communication. Below ground, an even more extensive communication network exists, mediated by fungi. This network, sometimes called the Wood Wide Web, connects the roots of different plants through fungal filaments called hyphae. The fungi involved are mycorrhizae, which form beneficial relationships with plant roots, helping plants absorb water and nutrients from soil in exchange for sugars from the plant. These mycorrhizal networks can span enormous areas, connecting dozens or even hundreds of different plants. And information can travel through these networks. When a plant is under attack, it can send signals through the mycorrhizal connections to neighboring plants. These signals might include chemical compounds that move through the fungal hyphae, or might even involve changes in the sugars the plant provides to the fungi, which the fungi then interpret as signals. Studies have shown that plants connected through mycorrhizal networks show coordinated defense responses. When one plant is attacked, connected plants that aren't being attacked can still show increased defensive chemistry and enhanced resistance to herbivores. The effect is more pronounced between plants of the same species, but can occur across species as well. Large, well-established trees sometimes appear to play keystone roles in these underground networks, serving as hubs that connect many younger or smaller plants. Some studies suggest that mature trees can even support the growth of younger trees through resource sharing via mycorrhizal networks, particularly when the younger trees are in shade and struggling to photosynthesize enough to meet their needs. If these networks also share defense signals, then the entire forest community might coordinate its defensive state, responding collectively to threats. There's also evidence of direct root-to-root communication without fungal intermediaries. When roots of different plants grow close together, they can exchange chemical signals directly through root exudates, substances that roots release into the surrounding soil. Some of these exudates include defensive compounds or signaling molecules that can be taken up by nearby plants and influence their defensive state. The chemicals involved in underground signaling are still being characterized, but they appear to include phenolic compounds, terpenoids, and various nitrogen-containing molecules. The soil environment is chemically complex, with thousands of different organic molecules present at any given time, making it challenging to identify which specific compounds serve as signals. But the biological effect is clear. Roots can influence the defensive state of neighboring plants through chemical communication. This interconnected view of plant communities has profound implications for ecology and agriculture. It suggests that plant diversity isn't just about having many different species present, but also about the functional connections between individuals. Disrupting mycorrhizal networks through practices like heavy tilling or fungicide use might not only harm individual plants' ability to acquire nutrients, but also break communication pathways that help coordinate community-wide defense. In natural ecosystems, the communication networks we're describing create resilient, adaptive communities. Information about herbivore outbreaks can spread through a forest stand or grassland, allowing plants to prepare defenses before they're directly attacked. Plants that are already stressed or damaged can potentially receive support from healthier neighbors through underground resource sharing. The community functions as more than just a collection of individual plants competing for resources. It operates as an interconnected system with emergent properties that arise from the interactions between its members. As research in this area continues, we are discovering additional layers of complexity. Some plants appear to be able to recognize chemical signals from close kin differently than signals from unrelated individuals, suggesting sophisticated signal discrimination. The volatile signals released by damaged plants can vary depending on the species of herbivore attacking them, potentially providing information not just about whether attack is happening, but about what kind of attacker is present. This web of communication, above ground through volatile compounds and below ground through fungal networks and root exudates, creates an information layer overlaying the physical structure of plant communities. Plants aren't isolated individuals stoically enduring whatever fate brings. They're networked organisms, sharing data about their environment and their condition with their neighbors. This fundamentally changes how we should think about plant communities and highlights yet another dimension of the sophisticated strategies plants use to persist in a world full of threats. Calling for backup. Indirect defenses. Not all plant defenses involve making themselves difficult or dangerous to eat. Some of the most elegant strategies involve recruiting allies, enlisting other organisms to defend the plant against herbivores. These indirect defenses represent a fascinating dimension of plant protection, one that highlights the complex ecological relationships that structure natural communities. By attracting the predators and parasites of herbivores, plants can reduce herbivore damage without bearing all the costs of defense themselves. The most widespread form of indirect defense involves volatile organic compounds that attract natural enemies of herbivores. When a plant is attacked by caterpillars, for instance, it releases a specific blend of VOCs into the air. These compounds don't just warn neighboring plants, as we discussed in the previous section. They also serve as a beacon that attracts parasitoid wasps, insects that lay their eggs in or on caterpillars. The wasp larvae then develop inside the caterpillar, eventually killing it. This phenomenon was first clearly demonstrated in corn plants. When caterpillars feed on corn leaves, the plants release volatile terpenoids that attract parasitoid wasps from considerable distances. Laboratory experiments showed that these wasps are specifically attracted to the volatiles released by damaged plants, not to undamaged plants. Furthermore, the blend of volatiles released varies depending on which species of caterpillar is feeding. The wasps can apparently use these different volatile signatures to locate their specific host species among potential prey. The sophistication of this system is remarkable. The plant must detect that it's being attacked, identify the specific herbivore attacking it, synthesize and release the appropriate volatile blend, and do all this while maintaining its other physiological functions. The parasitoid wasps, for their part, have evolved to recognize these plant-produced signals as indicators of prey location. This represents a tritrophic interaction, an ecological relationship spanning 3 levels of the food chain: plant, herbivore, and herbivore's predator. Different plant species produce different volatile blends when attacked, and sometimes even the same plant will produce different blends depending on which herbivore is attacking it. This specificity ensures that the attracted predators or parasites are ones likely to be effective against the actual herbivore present. It would do little good for a plant to attract predators of aphids when it's actually being attacked by caterpillars. The production and release of these attractive volatiles comes at a cost to the plant. It requires synthesizing new compounds and releasing them into the air rather than keeping them for other uses. But the cost appears to be much lower than the cost of producing large quantities of toxins or suffering extensive tissue loss to herbivores. By recruiting natural enemies, the plant gets protection at a bargain price. Some plants have taken this strategy even further by forming permanent associations with ant colonies. These ant-plant mutualisms represent some of the most remarkable defensive partnerships in nature. The plant provides food and shelter for the ants, and in return, the ants aggressively defend the plant against any herbivore that approaches. The classic example is the relationship between acacia trees and Pseudomyrmex ants in Central and South America. Certain acacia species have evolved hollow thorns that serve as homes for ant colonies. These thorns are often swollen and large, providing spacious chambers where ant queens can establish colonies and workers can rear their young. The tree doesn't stop at providing housing. It also feeds the ants through two mechanisms. First, these acacias produce specialized structures called Beltian bodies at the tips of their leaflets. These are tiny, nutrient-rich packages that ants harvest and feed to their larvae. Beltian bodies are costly for the tree to produce, but they ensure that the ant colony stays healthy and populous. Second, the trees produce extrafloral nectaries, nectar-producing glands located on leaves and stems rather than in flowers. This nectar provides carbohydrates to fuel the adult ant's activities. In exchange for this food and housing, the ant colonies mount fierce defenses. Any insect that tries to land on the tree is immediately attacked by patrolling ants. Larger herbivores, like browsing mammals, also face aggressive ant attacks, with ants swarming onto the animal's face and stinging repeatedly. The ants even clear vegetation around the base of the tree, removing vines and herbaceous plants that might compete with their host for light and nutrients. They essentially maintain a clear zone around their tree, gardening the environment to benefit their host. The specificity of these ant-plant mutualisms is remarkable. The ants depend entirely on the tree for food and shelter and have lost the ability to nest anywhere else. The trees, meanwhile, show reduced investment in chemical defenses compared to related species that don't host ants. They've essentially outsourced defense to their ant partners, reallocating resources that would have gone into making toxins or tough leaves into producing Beltian bodies and nectar instead. Similar mutualisms have evolved independently in other plant families. In Southeast Asia, macaranga trees host ant colonies in their hollow stems. In Africa, some species of whistling thorns house ant colonies and produce food bodies to sustain them. Each of these represents an independent evolutionary origin of this defensive strategy, suggesting that the benefits of ant protection are substantial enough to drive the evolution of these complex relationships repeatedly, Not all ant-plant interactions involve such intimate, species-specific partnerships. Many plants produce extrafloral nectar without providing housing for ants. This nectar attracts any ants in the area, and while the ants are feeding, they also patrol the plant and attack herbivores they encounter. This represents a less specialized form of ant defense, but it can still be highly effective. Studies comparing plants with and without extrafloral nectaries, or experimentally excluding ants from plants that normally have them, consistently show that ant-defended plants suffer less herbivore damage. The effect can be dramatic, with ant-defended plants losing only a fraction of the leaf tissue that undefended plants lose. For annual plants that must complete their entire life cycle in a single growing season, this difference can mean the difference between successful reproduction and failure. The timing of extrafloral nectar production is often strategic. Many plants produce more nectar during periods when herbivore pressure is highest, or when vulnerable young tissues are developing. This targeted resource allocation maximizes the benefits of ant defense while minimizing the costs of producing nectar when ants aren't needed as badly. Some plants even use chemical signals to recruit defenders. When damaged by herbivores, they release volatiles that attract not just parasitoids, but also predatory insects like lady beetles, lacewings, and predatory bugs. These predators consume herbivores directly, providing another layer of defense. The complexity of these interactions extends beyond simple attraction. Some plants appear to produce volatiles that not only attract predators, but also make the herbivores themselves more vulnerable. Certain VOCs can interfere with herbivore behavior, making them more likely to drop off the plant or become confused about where to feed. When a predator is hunting, these behavioral disruptions in prey can make successful predation more likely. Indirect defenses highlight the ecological embeddedness of plants. They don't exist in isolation, but are part of complex communities where species interact in multiple ways. The same compounds that might directly deter one herbivore can serve to attract the predators of another. The resources a plant invests in producing nectar for ants are resources it doesn't need to invest in producing toxins. The presence or absence of mutualist ants can influence which herbivores can successfully feed on a plant, which in turn shapes the evolution of herbivore populations. From a plant's perspective, indirect defenses offer several advantages. They can be effective against a broad range of herbivores, since predators and parasitoids typically attack multiple herbivore species. They allow plants to deploy targeted defenses, producing attractive volatiles only when actually attacked rather than maintaining constant chemical defenses. And they leverage the foraging abilities of mobile organisms, allowing tiny, volatile signals to recruit defenders from considerable distances. The discovery and documentation of these indirect defenses has been one of the most exciting developments in plant ecology over recent decades. It reveals plants as active participants in complex ecological networks using sophisticated strategies to manipulate the behavior of other organisms to their own benefit. When we look at a plant hosting an ant colony or releasing volatiles that attract parasitoid wasps, we're witnessing the results of millions of years of coevolution, the crafting of intricate ecological relationships that have been tested and refined across countless generations. movement as defense, dynamic responses. We typically think of plants as stationary organisms, rooted in place and unable to move in response to threats. While it's true that plants can't run from danger, many species can move in ways that reduce herbivore damage or make feeding more difficult for attackers. These thigmonastic movements in response to touch, represent another dimension of plant defense, one that combines physical and physiological mechanisms in fascinating ways. The most famous example of defensive plant movement is the sensitive plant, Mimosa pudica. If you brush against one of its leaves, it folds up rapidly, the leaflets closing together and the entire leaf drooping downward. The response is startlingly fast for a plant, occurring within seconds of touch. If you disturb the plant more vigorously, the response can cascade through the entire plant, with multiple leaves folding and drooping in sequence. The mechanism behind this rapid movement involves specialized structures at the base of the leaf and at the base of each leaflet called pulvini. These contain cells that rapidly change their water content in response to touch signals. When the plant detects mechanical stimulation, specific cells in the pulvini quickly lose water, becoming flaccid. This causes the cells on one side of the joint to lose a turgor pressure faster than cells on the other side, creating an imbalance that causes the leaf to fold or droop. The whole process is driven by osmotic phenomena, the controlled movement of water and ions across cell membranes. But why would a plant benefit from folding its leaves when touched? Several explanations have been proposed. The folded posture presents less surface area to a potential herbivore, essentially hiding the leaf away. It also changes the visual appearance of the plant, potentially making it look less like fresh foliage and more like a dead or diseased plant that wouldn't be worth eating. Some researchers have suggested that the sudden movement itself might startle small herbivores, causing them to drop off the plant. The drooping leaves might also physically dislodge insects that are trying to settle on the plant. There's evidence supporting multiple functions. Studies have shown that herbivorous insects do cause less damage to Mimosa pudica plants that can perform their touch response compared to plants experimentally treated to prevent the movement. The magnitude of the benefit varies with the type of herbivore, but overall, the touch response appears to provide genuine defensive value, enough to justify the energy cost of maintaining this system. And there are costs. The rapid movements require specialized structures and control mechanisms. Each time a leaf folds, it stops photosynthesizing until it reopens. If the plant is repeatedly disturbed, it can spend significant time with its leaves folded, reducing its total carbon gain. The sensitive plant seems to manage these costs through learning of a sort. If stimuli are repeated frequently without any actual damage occurring, the plant's response diminishes, a phenomenon called habituation. This prevents the plant from wasting energy responding to harmless touches from wind or passing animals. Mimosa pudica isn't alone in using rapid movements as defense. Several other plant species in different families have evolved similar touch-sensitive responses. The telegraph plant, Codariocalyx motorius, exhibits spontaneous leaflet movements even without touch, and these movements become more pronounced when the plant is disturbed. Various species in the genus Biophytum show folding responses similar to Mimosa. The convergent evolution of these mechanisms suggests that movement can indeed be an effective defensive strategy under the right circumstances. Venus flytraps represent perhaps the most dramatic example of plant movement, though in their case, the movement serves to capture prey rather than defend against herbivores. Still, the mechanisms are worth considering because they reveal the potential for rapid plant responses. The Venus flytrap's trap closes in about 1/10 of a second, fast enough to capture flying insects. This requires a sophisticated triggering mechanism involving sensitive trigger hairs that must be touched twice in quick succession to initiate closure, preventing the plant from wasting energy on false alarms like raindrops. The closure mechanism involves a combination of turgor changes and physical buckling of the leaf structure. The exact details are still being researched, but it's clear that electrical signals play a crucial role, propagating through the trap tissue when trigger hairs are touched. These electrical signals are remarkably similar to animal action potentials, though generated through different ion channels. Once closure is triggered, the trap snaps shut through a combination of rapid water movement between cells and the release of stored elastic energy in the leaf structure. While flytraps use their movement to capture insects for nutrition rather than to defend against herbivory, the underlying mechanisms, electrical signaling, rapid water movement, specialized tissues, are similar to those used in defensive movements. It demonstrates what plants are capable of when natural selection favors rapid responses. Some plants use movement defensively in subtler ways. Sunflowers and many other species track the sun across the sky through the day, a phenomenon called heliotropism. While this primarily serves to maximize light capture for photosynthesis, there's evidence it may also have defensive benefits. By orienting leaves to track the sun, plants can control their temperature. Cooler leaves might be less suitable for certain insect herbivores, particularly in cooler climates. The constant movement of sun tracking might also make it more difficult for insects to settle and feed comfortably. Vines and climbing plants show another form of defensive movement through their growth patterns. When they encounter support structures, they exhibit thigmotropism, growth responses to touch. Tendrils will wrap around anything they touch, allowing the plant to climb. But some studies suggest that these touch-responsive growth patterns might also help plants avoid certain herbivores or position themselves away from ground-level threats. Some grass species can regrow from their base when cut or grazed, a response that involves both developmental changes and movement of resources within the plant. While not movement in the sense of leaves folding, this represents an active response to damage that minimizes its impact. The grass redirects resources to basal meristems, growth regions at the ground level that are protected from further grazing, allowing rapid regrowth of lost tissue. The energy economics of movement as a defense are complex. Moving costs energy, but not moving might cost even more if it leads to greater herbivore damage. Plants that use movement defensively tend to be ones that experience frequent disturbance by herbivores or live in environments where herbivore pressure is high. The touch-sensitive responses are most common in tropical regions, perhaps because insect herbivores are more abundant and active year-round in tropical environments. Comparing the defensive strategies we've discussed, toxins, physical barriers, calling for help, and now movement, reveals the diversity of solutions plants have evolved to the same fundamental problem of surviving herbivory. Different strategies suit different lifestyles and environments. A long-lived tree can afford to invest heavily in permanent structures like thorns and thick bark. Annual plant might be better served by induced chemical defenses that can be activated when needed. A nitrogen-fixing plant with access to abundant nutrients might benefit from ant mutualism, producing expensive nectar to support ant colonies. A plant in an environment with frequent disturbance might benefit from rapid folding movements that startle or discourage herbivores. The existence of movement-based defenses reminds us that plants, despite lacking muscles and nervous systems, are not static organisms. They can sense their environment and respond to threats with remarkable speed. The fact that these responses are mediated by water movement, ion fluxes, and electrical signals rather than muscle contractions doesn't make them any less impressive as adaptations. They represent yet another example of the deep sophistication that has evolved in plants over hundreds of millions of years of living in a world where being eaten is an ever-present danger. When defense becomes offense, carnivorous plants. In perhaps the most dramatic reversal of the usual plant-herbivore relationship, some plants have evolved not just to defend themselves, but to actively hunt the very animals that might otherwise eat them. Carnivorous plants represent one of nature's most elegant solutions to survival in nutrient-poor environments. Unable to get sufficient nitrogen and other nutrients from their soil, these plants have turned predator, capturing and digesting insects and other small animals to supplement their nutrient intake. Carnivory in the plants has evolved independently, at least 6 times across different plant families, suggesting that under the right conditions, nutrient-poor soils and sufficient moisture, the benefits of capturing prey can outweigh the substantial costs of developing and maintaining trapping mechanisms. There are over 600 known species of carnivorous plants, found on every continent except Antarctica. They've developed diverse trapping strategies, each a marvel of evolutionary engineering. Let's begin with the Venus flytrap, Dionea muscipula, perhaps the most iconic carnivorous plant. Native to a small region of North and South Carolina, the flytrap uses a snap trap mechanism that closes with remarkable speed when triggered. The trap is actually a modified leaf, hinged along the midline. The inner surfaces are studded with 3 tiny trigger hairs on each lobe. When an insect touches one of these hairs twice in quick succession, or touches 2 different hairs within about 20 seconds, the trap snaps shut. The closure is one of the fastest movements in the plant kingdom, occurring in about 1/10 of a second. The mechanism involves a combination of turgor pressure changes and elastic energy stored in the trap's structure. When trigger hairs are touched, they generate electrical signals that propagate through the trap tissue. These signals trigger cells in the outer layers of the trap to rapidly lose water, changing their shape. The trap, which has been held in a curved position under tension, suddenly buckles inward like a loaded spring being released, the lobes snapping together. The sophistication doesn't end with capture. Once the trap closes, marginal teeth along the edges interlock, forming a cage. If the prey is small enough to escape through the gaps between teeth, the trap will reopen within a day or 2. But if a reasonably sized insect is trapped, Its continued struggles touching the trigger hairs signal to the plant that it has captured genuine prey rather than a false alarm like a raindrop or wind-blown debris. This continued stimulation causes the trap to seal more tightly, forming an airtight chamber. Glands on the inner surface then begin secreting digestive enzymes, proteases that break down proteins, phosphatases that liberate phosphorus from organic molecules, and other enzymes that dissolve insect tissues. Over the course of a week or more, the insect is digested, and the trap acts like an external stomach, absorbing the released nutrients directly through its inner surface. Once digestion is complete and nutrients have been absorbed, the trap reopens, ready for another capture. A single trap can close about 3 to 5 times before it can no longer function properly and dies back, while the plant continues growing new traps. This life cycle reflects the energy cost of trapping. Maintaining the trigger mechanism and producing digestive enzymes is expensive, and traps wear out. Pitcher plants represent a very different trapping strategy, the pitfall trap. Several plant families have independently evolved pitcher traps, including tropical pitcher plants of the genus Nepenthes, North American pitcher plants of the genus Sarracenia, and the Australian Albany pitcher plant. Despite their independent origins, these plants share common features because they solve the same problem. A pitcher trap is essentially a modified leaf shaped into a deep cavity filled with digestive fluid. The opening of the pitcher is typically adorned with nectar glands and bright colors that attract insects. The rim, called the peristome, is often slippery, causing insects to lose their footing. Below the rim, the inner walls are coated with waxy scales or downward-pointing hairs that make climbing out nearly impossible. Insects that fall in find themselves in a pool of enzyme-rich fluid at the bottom. The digestive fluid in pitcher plants is remarkable. It contains enzymes similar to those in animal digestive systems, proteases, chitinases that break down insect exoskeletons, and various other degradative enzymes. The fluid is acidic, with pH levels often below 3, comparable to stomach acid. This hostile chemical environment breaks down prey, and the pitcher absorbs the released nutrients through specialized cells lining its inner surface. Different pitcher plant species show interesting ecological variations. Some Nepenthes species have evolved enormous pitchers that can hold several liters of fluid and capture not just insects, but occasionally small vertebrates like frogs and rodents. Others have evolved more specialized niches. Nepenthes lowai, growing in nutrient-poor mountain forests of Borneo, has largely given up insect capture and instead catches the feces of tree shrews that visit the plant to feed on nectar secreted around the pitcher rim. This represents a sophisticated mutualism. The shrew gets food, and the plant gets nitrogen-rich feces deposited directly into its pitcher. Some pitcher plants host entire ecosystems within their fluid. Mosquito larvae, bacteria, and other organisms live in the digestive fluid, processing prey and releasing nutrients that the plant can then absorb. These pitcher plant communities have become model systems for studying ecological interactions in well-defined, contained environments. Sundews, in the genus Drosera, use yet another trapping strategy: sticky, adhesive traps. These plants are covered with tentacles, each tipped with a gland that secretes a sticky mucilage. When an insect lands on the leaf and touches these drops, it becomes stuck. The insect's struggles only cause it to contact more tentacles, becoming increasingly trapped. What makes sundews particularly fascinating is that their tentacles can move. When prey is captured, nearby tentacles slowly bend toward the prey, bringing more adhesive glands into contact and eventually wrapping the entire leaf around the trapped insect. This movement is slower than the Venus flytrap's snap, but still represents active capture behavior. The tentacles and leaf surface then secrete digestive enzymes, breaking down the prey over several days. The movement mechanism in sundews involves differential growth rates on the two sides of the tentacle and leaf, controlled by hormonal signals triggered by prey contact. Recent research has revealed that electrical signals and calcium waves propagate through the leaf when prey is captured, coordinating the movement of the multiple tentacles toward the capture site. Bladderworts, aquatic plants in the genus Utricularia, have evolved perhaps the most complex trapping mechanism of all. They produce tiny bladder-like structures, each about the size of a pinhead, that function as suction traps. The bladder sets itself like a trap by pumping water out, creating negative pressure inside. The entrance is sealed by a watertight door with trigger hairs protruding from it. When a small aquatic organism like a water flea touches the trigger hairs, the door springs open, water rushes into the bladder due to the pressure differential, and the prey is sucked inside along with the water. The entire process takes about 1/1000th of a second, making it one of the fastest movements known in any organism, plant or animal. Once inside, the prey is digested by enzymes, and the bladder resets itself for another capture. The sophistication of carnivorous plants extends beyond their trapping mechanisms to their overall ecology and evolution. These plants are photosynthetic and produce their own energy through photosynthesis, like other plants. The captured prey provides supplemental nutrients, primarily nitrogen and phosphorus, that are scarce in the boggy, acidic soils where most carnivorous plants live. Experiments comparing carnivorous plants-fed insects to those without prey show that fed plants grow faster and produce more offspring, demonstrating the nutritional benefit of carnivory. The environments where carnivorous plants thrive are typically characterized by high moisture, high light, and nutrient-poor soils. Bogs, swamps, and rocky seeps often lack sufficient nitrogen because microbial decomposition is slow in acidic, waterlogged conditions. By capturing insects, carnivorous plants access a nutrient source unavailable to competing plants. This allows them to dominate in environments where other plants struggle. The evolution of carnivory requires numerous adaptations working together. The plant needs structures that can attract, trap, and retain prey. It needs digestive enzymes it can deploy outside its normal cellular environment. It needs the ability to absorb nutrients through leaf surfaces rather than roots. And ideally, it needs mechanisms to distinguish prey from false triggers, avoiding wasted effort on raindrops or wind-blown debris. These requirements explain why carnivory has evolved relatively few times despite its advantages. The developmental and metabolic changes required are substantial. many intermediate steps would need to occur, each providing some benefit that natural selection could favor. But once the full carnivorous syndrome is in place, it can be highly successful, as evidenced by the diversity of carnivorous plants and their ability to thrive in some of Earth's most challenging habitats. When we consider carnivorous plants in the context of plant defenses, they represent a remarkable pivot. The same traits that some plants use to defend themselves, toxic chemicals, sticky secretions, movement in response to touch, have been co-opted by carnivorous plants as tools for predation. The enzymes that might digest herbivore gut contents in a defensive plant become tools for digesting the herbivore itself in a carnivorous plant. The sticky compounds that trap insects in defensive trichomes become the adhesive of sundew glands. The rapid movements that fold leaves away from herbivores become the snap of a flytrap. This reveals the evolutionary flexibility of plants, their ability to modify existing traits for new functions. Carnivorous plants haven't evolved entirely new molecular machinery. Instead, they've repurposed and enhanced systems that already existed, directing them toward a new function: active predation. It's a testament to the power of natural selection to shape complex adaptations, and a reminder that the line between defense and offense can sometimes blur in fascinating ways. Cellular and molecular defenses. While we can see physical barriers like thorns and observe dramatic behaviors like Venus flytraps snapping shut, many of the most sophisticated plant defenses operate at microscopic scales, invisible to the naked eye. At the cellular and molecular level, plants wage chemical warfare with invaders, deploying an arsenal of defensive mechanisms that rival the immune systems of animals in their complexity and effectiveness. Understanding these microscopic battles reveals just how active and responsive plants really are to threats. When a pathogen, a disease-causing bacterium, fungus, or virus, attempts to infect a plant, it must first overcome the physical barrier of the cell wall. This wall, composed primarily of cellulose, provides structural support and acts as the first line of defense. But the cell wall is more than passive armor. It's a dynamic structure that plants can modify rapidly in response to attack, reinforcing it with additional materials to make it even harder for pathogens to penetrate. Upon detecting a pathogen, plant cells can deposit extra layers of materials into their cell walls. Calhus, a complex sugar polymer, is rapidly synthesized and deposited, creating thick barriers that strengthen the wall and plug connections between cells, preventing pathogen spread. Lignin, the polymer that makes wood hard, can be deposited in cell walls within hours of infection, creating a lignin barrier that most pathogens cannot penetrate. Suberin, a waxy, waterproof substance, creates additional barriers that resist both physical penetration and water movement, which many pathogens need for infection. But perhaps the most dramatic cellular defense is the hypersensitive response. When plant cells detect a pathogen, they can undergo programmed cell death, sacrificing themselves to prevent infection from spreading. This suicide mission serves several purposes. First, it deprives the pathogen of living host tissue to colonize. Many pathogens can only grow in living cells, so when those cells die, the infection stalls. Second, cell death releases antimicrobial compounds that were stored safely in living cells but become toxic to pathogens when released. Third, the dead tissue forms a barrier that physically contains the infection. The hypersensitive response looks dramatic under a microscope or to the naked eye. Infected areas turn brown and die, often surrounded by a ring of reinforced cells that seal off the infection zone. To the plant, this localized cell death is a small price to pay for preventing systemic infection that could kill the entire organism. It's analogous to cutting off a finger to prevent gangrene from spreading. A drastic measure, but potentially life-saving. The detection system that triggers these responses is remarkably sophisticated. Plants use pattern recognition receptors on their cell membranes that can detect molecules associated with pathogens. These pathogen-associated molecular patterns, PAMPs, include components like fungal cell wall fragments, bacterial flagellin proteins, and various microbial enzymes. When these receptors bind their target molecules, they trigger a cascade of cellular responses. Within seconds of PAMP detection, calcium ions flood into plant cells, creating one of the earliest signals of infection. This calcium influx triggers downstream signaling cascades involving protein kinases, enzymes that activate other proteins by attaching phosphate groups to them. These cascades amplify the initial signal, ultimately activating defense genes throughout the cell and even in neighboring cells. Reactive oxygen species, ROS, form another crucial part of the early defense response. Plants generate superoxide radicals, hydrogen peroxide, and other highly reactive molecules at infection sites. At low concentrations, ROS act as signaling molecules, helping coordinate defense responses. At high concentrations, they are directly toxic to pathogens, damaging their cell membranes, proteins, and DNA. Plants must carefully control ROS production to harm pathogens without causing excessive damage to their own tissues. One of the most important classes of defensive proteins produced in response to infection are the pathogenesis-related proteins, or PR proteins. Scientists have identified multiple families of PR proteins, each with different defensive functions. Some PR proteins attack pathogen cell walls directly. Glucanases break down fungal cell wall components, literally dissolving the structural framework of invading fungi. Chitaeases attack chitin, another major component of fungal cell walls. By producing multiple enzymes that attack different cell wall components, plants ensure that fungi cannot easily evolve resistance to all of them. Other PR proteins have antimicrobial properties through different mechanisms. Some form pores in pathogen cell membranes, causing them to leak and die. Others bind to essential nutrients like iron, making them unavailable to pathogens. Still others seem to act as signals, alerting other parts of the plant to the ongoing infection and priming distant tissues for possible attack. Phytoalexins represent another crucial molecular defense. These are antimicrobial compounds that plants synthesize rapidly in response to infection. Unlike constitutive defensive chemicals that are always present, phytoalexins are produced on demand. different plant families produce different phytoalexins, reflecting their diverse evolutionary histories and the different pathogens they've evolved to resist. In potato and tomato plants, phytoalexins include compounds like rischitin and phytuberin. In beans and other legumes, phaseolin and other isoflavonoids serve as phytoalexins. In the crucifer family, compounds derived from sulfur-containing amino acids act as phytoalexins. What these diverse compounds share is their ability to accumulate rapidly to high concentrations at infection sites and their antimicrobial properties. The synthesis of phytoalexins is tightly regulated. Genes encoding the enzymes that make phytoalexins are normally expressed at very low levels or not at all. Upon infection, signaling cascades activate transcription factors, proteins that bind to DNA and turn genes on. These transcription factors ramp up expression of the entire pathway of enzymes needed to synthesize phytoalexins, often increasing their abundance thousands of fold within hours. This allows plants to produce phytoalexins rapidly without maintaining the metabolic burden of constantly producing them. The phenomenon of systemic acquired resistance, SR, which we touched on earlier, represents a whole plant-level molecular defense strategy. When a plant successfully fights off an infection in one location, it develops enhanced resistance throughout its entire body that can last for weeks or months. This is mediated by signaling molecules, particularly salicylic acid, that move through the plant's vascular system from the infection site. When salicylic acid levels rise in distant, uninfected tissues, they trigger expression of PR proteins and other defensive mechanisms throughout the plant. This primes the plant's defenses, making it respond more quickly and strongly to subsequent infections. Some of the defensive proteins are produced at low levels immediately, providing baseline protection. Others remain at low levels but with their genes poised for rapid activation if infection occurs. The plant essentially shifts into a heightened state of alert, ready to mount strong defenses at a moment's notice. RNA silencing represents yet another molecular defense mechanism, particularly important against viral pathogens. When a virus infects a plant cell, it produces double-stranded RNA as part of its replication process. Plants recognize this double-stranded RNA as foreign, since their own genes produce primarily single-stranded RNA, and target it for destruction using specialized protein complexes. These complexes cut the viral RNA into small pieces, which then guide the destruction of additional viral RNA with matching sequences. This RNA silencing system is so effective that many plant viruses have evolved proteins that suppress it. The evolutionary arms race between plants and viruses has led to increasingly sophisticated silencing systems in plants and increasingly effective suppressor proteins in viruses. The molecular details of these interactions are complex, but the bottom line is that plants have evolved a sophisticated system for detecting and destroying viral genetic material. At an even more fundamental level, plants deploy molecular defenses based on their recognition of self versus non-self. Through systems of receptor proteins and signaling molecules, plants can detect when they're being colonized by organisms that don't belong. This discrimination allows them to maintain beneficial associations with symbiotic fungi and bacteria, while mounting strong defenses against pathogens. The costs of maintaining these molecular defense systems are substantial. Producing defensive proteins requires amino acids, and synthesizing them requires energy. Maintaining the surveillance systems that detect pathogens requires constitutive expression of receptor proteins and signaling molecules. The hypersensitive response involves deliberately killing one's own cells. Synthesizing phytoalexins diverts metabolic resources from growth and reproduction. Yet these costs are clearly worth it, as evidenced by the ubiquity of these systems across the plant kingdom. Every plant studied in detail has been found to possess sophisticated molecular defenses. The specific details vary. Different plants use different phytoalexins, different PR proteins, different signaling pathways, but the overall strategy is universal. Detect pathogens early, respond rapidly and specifically, and deploy multi-layered defenses that make infection difficult. When we zoom out from these molecular details to consider their ecological implications, we see that the invisible battles occurring in every leaf and stem are just as important to plant survival as the more visible defenses we can see and touch. A plant that successfully resists fungal infection through molecular defenses will outcompete a susceptible neighbor. One that quickly detects and responds to viral infection will suffer less damage than one with slower recognition systems. The molecular sophistication of plant defenses reminds us that plants are not simple organisms. They're complex living systems with sensing capabilities, signal processing, coordinated responses, and molecular machinery rivaling that of animals. The fact that these systems operate through biochemistry rather than neurons doesn't make them any less impressive as adaptations or any less crucial to understanding how plants survive in a world full of threats, both large and small, both visible and microscopic. Costs and trade-offs. Throughout this journey into plant defenses, we've encountered numerous references to costs, the price plants pay for being well defended. Now, let's examine these costs more carefully and understand the fundamental trade-offs that shape defensive strategies. Every organism has finite resources, finite energy from photosynthesis, finite nutrients from the soil, finite time in a growing season. Resources allocated to defense cannot be used for growth, reproduction, or other functions. This creates evolutionary tensions that have shaped the diverse defensive strategies we see across the plant kingdom. The most direct cost of defense is the metabolic expense of producing defensive compounds and structures. Manufacturing alkaloids, terpenes, and other secondary metabolites requires energy and often specific nutrients, particularly nitrogen. A plant producing high concentrations of defensive chemicals is a plant channeling resources away from other uses. Studies comparing plant varieties that differ in defensive chemistry consistently show that better defended varieties grow more slowly when herbivores are absent, indicating a real cost to defense. Physical defenses like thorns and trichomes also carry costs. Building these structures requires carbon for structural materials, nitrogen for proteins, and other nutrients. Thorns represent woody tissue that could have been leaves capturing light for photosynthesis. Dense trichome coverage might deter herbivores, but also creates a boundary layer of still air around leaves that can impede gas exchange. potentially reducing photosynthetic efficiency. The costs of induced defenses are more complex. While these defenses are only deployed when needed, thereby avoiding constant maintenance costs, the detection and signaling systems required for induction still carry costs. Maintaining receptor proteins, signal transduction pathways, and the genetic machinery for rapid defense gene activation all require resources. Moreover, mounting an induced response itself is costly. When a plant shifts into defense mode after attack, producing protease inhibitors, phytoalexins, and other defensive proteins, growth often slows or even stops temporarily. Studies measuring the fitness costs of induced responses have found variable but generally significant impacts. Plants that are induced to produce defenses by experimental herbivory or by application of defense-inducing hormones typically produce fewer seeds than uninduced plants, reflecting the trade-off between defense and reproduction. The magnitude of this cost varies with environmental conditions. When resources are abundant, plants can better afford to defend themselves. When resources are scarce, defense competes more directly with other needs. This leads to predictions about when different defensive strategies should evolve. The optimal defense theory proposes that plants should allocate defenses where they'll provide the most benefit relative to their cost. Young leaves, which have high value to the plant and high remaining lifetime, should be better defended than old leaves nearing senescence. Reproductive structures should be highly defended because their loss directly reduces fitness. Plant parts that are most apparent to herbivores or most vulnerable to attack should receive greater defensive investment. These predictions are generally supported by observations. Young leaves typically contain higher concentrations of defensive chemicals than mature leaves. Seeds are often extremely well defended, containing toxins at concentrations that would be metabolically expensive to maintain throughout all plant tissue. Flowers often produce compounds specifically to deter herbivores while attracting pollinators, a delicate balancing act that requires sophisticated chemistry. The growth differentiation balance hypothesis offers another framework for understanding defense allocation. This hypothesis proposes that rapidly growing tissue is inherently less well defended because the physiological state that promotes growth, high nutrient availability, active cell division, metabolic focus on building new tissue, is incompatible with the physiological state that promotes defense. Secondary metabolism, production of non-essential compounds. Plants must therefore balance growth against defense, and this balance shifts depending on conditions. Under favorable conditions with abundant resources and low herbivore pressure, plants should prioritize growth, accepting higher vulnerability to herbivores because rapid growth allows them to recover from damage and outcompete neighbors. Under stressful conditions or high herbivore pressure, plants should prioritize defense, even at the cost of slower growth, because the alternative is losing too much tissue to reproduce successfully. Environmental factors strongly influence this growth-defense balance. Water stress, nutrient limitation, extreme temperatures, and other stresses all affect a plant's ability to both grow and defend itself. Often, stressed plants become more vulnerable to herbivores, not because they reduce defenses, but because they can't mount as strong an induced response when attacked. The resources simply aren't available for both stress tolerance and defense. This creates unfortunate feedback loops in nature and agriculture. A drought-stressed crop plant, already struggling to maintain basic functions, may be less able to defend itself against insect pests. The pest damage further stresses the plant, reducing its growth and yield. Agricultural management practices that support plant health, adequate water, balanced nutrition, disease control, help plants maintain effective defenses, breaking these negative feedback cycles. The concept of ecological costs adds another dimension to understanding defense trade-offs. Even if a plant can afford the metabolic cost of producing defenses, there might be ecological costs related to how those defenses affect interactions with other organisms. For instance, a plant producing volatile compounds to attract predators of herbivores might inadvertently attract other insects that aren't beneficial. Chemical defenses might deter not just herbivores but also pollinators, if defensive compounds are present in flowers. Some defensive strategies can even attract specialized herbivores. When plants produce specific defensive chemicals, herbivores can evolve to specialize on those plants, using the defensive compounds as host recognition cues. Over time, what was once an effective defense becomes a dinner bell for adapted specialists. This creates ongoing selection for plants to modify their defenses, contributing to the evolutionary arms race we discussed earlier. The choice between constitutive and induced defenses represents a fundamental trade-off with deep evolutionary implications. Constitutive defenses provide immediate protection but cost energy, even when threats are absent. Induced defenses save energy when threats are low, but leave plants vulnerable during the lag time between attack detection and full defense deployment. Which strategy evolves depends on the predictability and intensity of herbivore pressure. In environments where herbivore pressure is consistently high, constitutive defenses are favored. The cost of maintaining them is justified by their constant use. In environments where herbivore pressure is unpredictable or seasonal, induced defenses are favored. Plants can save resources during safe periods and deploy defenses only when needed. Long-lived plants, which must survive many years and unpredictable conditions, often combine both strategies, maintaining baseline constitutive defenses while retaining the ability to induce additional defenses when attacked. Plant lifespan also influences defense strategies. Annual plants, which must complete their entire life cycle in a single growing season, often invest less in defense than perennial plants. An annual that suffers moderate herbivore damage can potentially tolerate it, growing rapidly to produce seeds before dying. A perennial plant that loses significant tissue faces years of reduced growth and may not survive to reproduce. Perennials therefore tend to invest more heavily in defense, particularly long-lasting physical and chemical barriers. Resource availability in the environment shapes defense evolution too. In nutrient-poor environments, losing tissue to herbivores is especially costly because replacing it is difficult. Plants in these environments, think of the nutrient-poor bogs where carnivorous plants thrive, often invest heavily in defense. In nutrient-rich environments, tissue is easier to replace, so rapid growth and tolerance of herbivory can be viable strategies even with less investment in defense. These patterns create predictable variation in plant defenses across ecological gradients. Moving from fertile valley bottoms to nutrient-poor mountaintops, we often see plants becoming progressively more defended. Moving from temperate zones where herbivory is seasonal to tropical zones where herbivory is year-round, we see increased investment in constitutive defenses. These aren't absolute rules. Every environment contains variation and exceptions, but they reveal general principles about how costs and benefits shape defense evolution. Understanding these costs and trade-offs has practical implications for agriculture and conservation. Plant breeders developing crop varieties face decisions about how much defensive capacity to maintain. Highly defended varieties might resist pests better, but yield less under pest-free conditions. Less defended varieties might yield more, but require more pest management. Finding the right balance depends on the specific crop, growing region, and farming practices. In restoration and conservation, understanding defense trade-offs helps predict how plants will respond to changing conditions. If herbivore pressure increases due to loss of predators or introduction of invasive herbivores, how will native plants respond? Plants with high constitutive defenses might better resist new threats. Plants relying on induced defenses might struggle if they haven't evolved to recognize the new herbivore's attack signatures. As we step back from this examination of costs and trade-offs, we see that defense isn't just about having the biggest thorns or the deadliest toxins. It's about making strategic decisions with limited resources, balancing competing demands, and adjusting strategies based on conditions. Natural selection has been running these optimization calculations for millions of years, generating the stunning diversity of defensive strategies we observe today. Every plant we encounter represents a solution to the defense problem that worked well enough, in that species' particular circumstances, to persist across generations. Conclusion. As we near the end of our journey through the hidden world of plant defenses, take a moment to let your awareness settle. Breathe slowly. Feel the weight of everything we've explored together gently resting in your mind like leaves settling onto still water. We've traveled from ancient evolutionary arms races through chemical laboratories hidden in leaf tissue, from underground communication networks to traps that snap shut faster than an eye can blink. And through it all, a single truth has emerged. Plants are not passive victims, but sophisticated defenders, evolved over hundreds of millions of years to protect themselves with remarkable ingenuity. Think about what this means for your daily experience of the world. That garden you walk past, the park where you rest on summer afternoons, the forest where you hike. These spaces are alive with invisible warfare. Every blade of grass contains silica crystals sharp as glass at the microscopic level. Every rose thorn represents a branch that chose protection over growth. The bitter taste of certain leaves, the sticky sap that wells up from a broken stem, the thorny barriers around fruit, all of these are messaged in a language far older than words. I am defended. I am not easy prey. Choose someone else. The sophistication we've witnessed should inspire a deeper respect for plants. They can't flee from danger or fight with claws and teeth. Rooted in place, they must face every threat exactly where they stand. Yet they've developed sensing systems that detect danger, communication networks that warn neighbors, chemical weapons that rival any laboratory, alliances with insects that serve as bodyguards, and in some cases, the ability to turn predator themselves. They've optimized their defense spending like strategic economists, deciding when to invest in security and when to focus on growth. They've learned, in an evolutionary sense, when to be toxic, when to be tough, when to call for help, and when to simply tolerate damage and grow back. These strategies reflect deep intelligence, though not consciousness as we understand it. It's the intelligence of systems refined over eons, tested against countless challenges, shaped by the unforgiving mathematics of survival. Every defensive mechanism we've discussed exists because the plants that possessed it lived more successfully than those that didn't. Natural selection has been running experiments longer than we can truly comprehend, and the results surround us in every plant we encounter. Consider how this knowledge might change your relationship with plants. That stinging nettle that caught your arm during a hike? It wasn't attacking you maliciously. You triggered its defense system, those remarkable hollow hairs functioning exactly as evolution designed them. The bitter taste of dandelion greens? That's chemical warfare compounds meant to discourage herbivores, and your taste buds are correctly identifying them as potentially harmful. The fuzzy coating on some leaves that makes them feel soft. That might be a trichome barrier making it difficult for tiny insects to reach the leaf surface. Even in agriculture, which seems so distant from wild nature, these principles apply. Crop plants are still plants, still facing threats from insects and diseases, still deploying the same basic defensive strategies their wild ancestors used. The difference is that we've often bred some of those defenses out, selecting for taste and productivity over protection, then using pesticides to provide the security the plants can no longer fully generate themselves. Understanding plant defenses might help us develop agricultural systems that work with these natural mechanisms rather than replacing them, supporting crops' own protective abilities while reducing our reliance on chemical inputs. The future holds exciting possibilities for this field. As we develop better tools for studying molecular processes in plants, we'll undoubtedly discover defensive mechanisms we haven't yet imagined. The plant kingdom is vast, with hundreds of thousands of species, and we've studied only a tiny fraction in detail. Each one likely contains unique solutions to the defense problem, novel compounds and strategies that could teach us new things about how life adapts to challenges. There are practical applications too. Many of our medicines come from plant defensive compounds. The salicylic acid pathway involved in plant disease resistance gave us aspirin. Taxol, a cancer chemotherapy drug, comes from the bark of Pacific yew trees, where it serves as a defense against pathogens. As we discover new plant defensive compounds, we might find new medicines, new natural pesticides, new solutions to human problems hidden in the chemistry plants evolved for their own survival. But perhaps the most important thing we can take from this exploration is simply a sense of wonder. The world is far stranger and more marvelous than casual observation suggests. Plants are not static decorations, but dynamic organisms engaged in constant interaction with their environment. They sense, they respond, they communicate, they make strategic decisions about resource allocation. They're players in ecological dramas that span continents and geological ages. They're worthy of our attention, our respect, and our curiosity. So, the next time you're in nature, take a moment to look more closely. Touch a leaf and wonder about the chemicals in its tissue, the structures on its surface, the cellular machinery ready to spring into action if that touch becomes a bite. Smell the fragrance of crushed herb leaves and consider that you might be detecting volatile signals meant for other organisms entirely. Watch an ant crawling on a plant stem and consider whether it's just passing through or actually patrolling its host in exchange for food and shelter. Notice thorns, sticky saps, tough textures, bitter tastes, each one a window into the hidden world of plant defense. These observations won't diminish the beauty of nature. If anything, they enhance it. Understanding the mechanisms behind a flower's scent or a tree's thorns doesn't make them less lovely. It adds layers of meaning, connecting each individual observation to vast networks of ecological and evolutionary relationships. Beauty and knowledge aren't opposed. They're complementary, each enriching the other. Before we part, if you've enjoyed this quiet exploration of plant defenses, I encourage you to like this video and subscribe to the Sleepy Science Society. Your support helps these peaceful journeys through science reach others who might appreciate them. Consider sharing your thoughts in the comments about which plant defense mechanisms you found most fascinating, or which ones you've noticed in your own environment. We're all learning together, all discovering new wonders in the world around us. And now, as we close, let yourself settle fully into comfort. Your breathing can slow. Your muscles can relax. Everything we've discussed tonight will still be there tomorrow, encoded in the plants you pass, the gardens you tend, the forests you visit. The knowledge settles into your mind like sediment layers, some facts retained clearly, others creating a general sense of understanding and appreciation. Both are valuable. Both are gifts from this time we've spent together exploring the secret warfare of the botanical world. The night is here to hold you gently. The defenses of plants continue their ancient vigil, thorns standing guard, toxins waiting in cell vacuoles, communication networks humming with molecular messages. But you can rest. You've earned this peace. Let your eyes close if they wish. Let thoughts drift like leaves on a slow stream. Let the wonder of plant defenses fade into a warm glow at the edges of awareness as sleep draws near. Thank you for joining me on this journey. Until next time, sleep well, dream peacefully, and may you always find wonder in the living world around you. Good night.